challenges.
Just to comment on that, and interesting as on the outside of looking in from the community perspective with data, right? So, and we've come across this in many different study plannings currently of that. A lot of it is diagnosis by exclusion for HIE because the medical records and the coding is not great. And that's problematic. So working towards, I think this is where advocacy, has been more effective, hopefully, and will continue to be, where we're, collaborating with like the Newborn Brain Society and the Cerebral Palsy Research Network and things like that to figure out what these common data elements should be. So that way people have a better idea. I mean, there's very few, I think, other diagnoses that get lumped like HIE does. And so we've had some issues with, just the landscape where people are coming to me and saying, where's the data? And I'm like, that's a great question, so that's why we're working collaboratively with everyone on that.
Okay. Jason thought I was going to get out away from without questions, but Jason, no. This is for the three middle panelists. Jason, if you can start us out. Obviously, biomarkers are really going to be an important consideration as we design clinical trials, in neonates. And obviously, trial duration is a huge factor as we think about this. So there's a bit of, adding supportive evidence, but also within a trial, but also giving back, right, in some ways of the future use cases and such. For those of you that are designing trials, what has been your approach for considering biomarkers? If there's ones you feel comfortable discussing, please feel free to add those. Why are they important to you all, and how are you thinking through them as you begin to think about this related to like maybe even mechanism of disease, related to mechanism of your drug? Thoughts, Jason? You're gonna
just to start, yeah. So our thought we're developing something, a treatment for white matter injury, and so our big challenge is funding. It's very, very expensive to run these studies and so one of the challenges with white matter injury is there's no good diagnostic, and so we do know that the incidence of white matter is the more premature the baby, the higher the rate, and so that's one indicator. But it would be great to have some sort of diagnostic or marker indicator that would indicate who had white matter injury and who didn't. And so there's a couple of things that we're looking at there, a couple of markers that we're looking at there. A paper came out last year on subventricular zone echogenicity that we're looking at that may be an indicator that there is white matter injury there. And then the benefit of having kind of a diagnostic marker is that it's kind of a win-win-win. It's a win for the patients because we then don't treat babies that don't have white matter injuries. We only treat those babies that have it. And then it allows us to have a smaller trial, a faster trial, and which again makes it easier to fundraise. And so I think kind of breaking this out, we have kind of diagnostic biomarkers, which we're looking at. And then there's kind of the other end, it's in the efficacy biomarkers. And again, just from an investor's perspective, they can't wait 8 years. They can't wait 13 years. We can't even get them to wait 2 years. So we're looking for those earlier signals. And again, sometimes you have to have safety data that goes out long enough, but it's trying to get that early signal so that we can show. And sometimes it may not, and we've kind of concluded that it may not hit kind of maybe regulatory kind of levels, but it'll be enough to give a signal to our investors to say, hey, let's go ahead and fund that next study that will then be at a regulator kind of level of efficacy. So it's kind of this stepwise approach, but biomarkers are an important part of that.
So you're saying both sort of a diagnostic biomarker, but are also perhaps a progression biomarker or something you could use for futility? Is that what I heard?
Yeah.
Okay.
Yep. Yeah.
Okay.
So I'm pretty sure I speak for the industry members in the audience that the two biggest headwinds we face are time and risk. And it has to do with investors. It has to do with the fact that we're charged with coming up with a new treatment and it's super expensive. And, with no clearly defined pathway for HIE, for example, historically trials have been designed with more adult-like measures of how they feel, function, and survive. So understanding that, adults, of course, you can see immediately what function someone has lost with a stroke, for example, but, and they can tell you how they feel. The babies are extremely disadvantaged with this approach. So this means, obviously, they haven't got a lot of function to measure without waiting all the years for the Bailey exam or 5 years. I'm, shuddering at the thought of trying to get someone to fund a study that would take 5 or 8 years. It's difficult. But the good news is, biomarkers, like unvalidated biomarker, or reasonably likely and a surrogate is, I mean, game changing, right? So for us, if we can get to that place, and this is why INC and NBIC is really important, these biomarkers really are going to, change the game. But the good news is the technology's here. MRI scores, EEG traces, other parameters are being measured every single day in the NICU, and they're being used to guide treatment decisions. Our KOLs and investigators are telling us this is what we use to determine the path, that as best we can, how to treat these babies. So these are already there. But what we have to do is put them into action as approvable endpoints, and that's what's going to help us conduct faster trials and can help the agency to make faster decisions on our behalf and get these treatments out for babies and families that are needed.
Did you have something you want to add?
So a question about biomarkers.
Sure.
Then. So what I'm hearing is you're focusing more on signals, brain signals, and or imaging techniques. What about when we talk about biomarkers? And obviously, I think more in the serum, the CNS, urine. I know there have been several biomarkers that have been studied over the years. Have any of them really risen to the top in terms of being able to determine how much injury there is, or actually predictive in terms of outcomes? And could those be used ultimately to support let's say a regulatory filing or evidence of effectiveness? Because waiting 3, 5, 8 years, sometimes for an industry player is sometimes it's a non-starter.
Yeah, that's a beautiful question for us because we the biomarkers that are most important to us have to do with inflammation, the inflammatory signals. So our drug is an anti-inflammatory. So we've been studying different biomarkers of inflammation. And so MPO is one of the markers that is the myeloperoxidase is one of the downstream markers that we can see the extent to which inflammation is occurring, and to the extent that we all believe inflammation is part of the what causes the brain injury in neonates. We can say that's really high. We can see there's inflammation going on, and then we can see how the drug affects that the MPO levels, and that's become a really important biomarker. And we've gone ahead and paid as a company to have it validated so that everyone can use that biomarker. And it's because we want to be the leader here, and we would like other people to be able to have access to that biomarker. So there are companies now who can run that assay in a validated manner. That's one of the ways. Well, here we go.
So I'll say also from my prior academic hat, I've done a fair amount of biomarker studies. A lot of these are small, either single or multi-center studies. We've also and this is a plug for all future clinical trials to include biomarkers in there as secondary endpoints or as exploratory aims in their trials, because I think that's the only way we're going to be able to quote unquote validate any of these candidate markers that are out there, markers of inflammation, brain specific markers, and in this area that we've been talking about in HIE, there was sub-studies of the most recent big clinical trial of erythropoietin that evaluated biomarkers as well. So there's always these, this is the signal amongst the noise. But as far as anything that's gotten to the point of, that you would rest a regulatory decision on, I think they're more in the phase of, PD type markers. So if you think that your drug has an anti-inflammatory effect, and you can show that these markers are a part of that in the early phases of development, I think that's where we are right now with blood-based biomarkers to support those types of things to move on. But I don't think we're at the phase where any primary blood-based biomarker would be where we would rest our hat as far as approving a drug.
Right. And to be clear, we're using that as a PD marker more than anything. I mean, as far as endpoints go, we look more at the traditional, the MRI as a measure of brain injury. And there's been so much publication, so much work done on that. And so, the hardest part about that is the variability in the associated with, the equipment and all those things. But I think if you talk to anyone who is an expert in the Bailey assessment, they will tell you that that's actually fraught with much more variability than any other sort of assessment, because you have to rely on the cooperation of a 2-year-old. And if anybody's got a 2-year-old and you know how it is to get them to cooperate for 90 minutes, that's pretty challenging, even for the most astute pediatric neurologist. So, but with the MRI and the EEG, you can take out as much of the variability as possible using contractors. We have people who are most expert at capturing and evaluating MRIs and EEGs. They assist with training, making sure all the equipment is uniform across all the sites. They do oversight. They even help with logistical things like uploading and downloading images. So, this is the way to wring out as much variability as we can. But again, the human assessment is the hardest part, right? That's
one of the key issues with the biomarkers, the serum CSF biomarkers, is it doesn't tell you about the region, and that's what MRI does for you, right? So you could have a small, in neurology is like real estate, location is everything. So you could have a small injury in a key area of the brain that is really produces, a lot of disability. And you could have a large injury in another area of the brain that produces pretty minor impairments. And so that's where I think the serum and CSF biomarkers have not been so valuable compared to something that has more regional and potentially even temporal specificity if you get more than one scan. So getting regional, honestly, you'll get more sort of temporal information from EEG, because you get several days of EEG monitoring that give you sort of not only where did you start from, but how quickly did you recover? And then MRI in terms of location and severity of any injury. So that's why people have focused on those rather over the serum and CSF markers to a large degree.
Right. And then the real way to help with the variability in the imaging and interpretation is to have central reading. And that's another thing that we've employed in the clinical trial, is to use central readers that can, be consistent as possible across all the measures. So that we don't know any other better way.
Really, one day we have automated reading, right?
Well,
that's what I
that's
think
right.
we would really
AI,
rather
right?
have.
Hopefully that'll be here one day. And there's new little companies that we're, working with to help them develop their software in that regard. But we're not there yet. So we're going to rely on the central readers and so forth. So.
I guess where it sits as well is, and I'm preaching to the choir, is the heterogeneity of the presentation of the infants with HIE. And it's difficult to sort of hang your hat on a single, biomarker, whether that's imaging related or blood related. And one of the ways that we're going down is, we've spoken about comparing apples to apples and oranges to oranges, but with the landscape of machine learning, we're now being able to evaluate the fruit salad and that the heterogeneity of HIE is, and it's looking at sort of a stepwise assessment of that, whether we're looking at, EEG within the first sort of 24 hours to look at seizure burden, followed up by then MRI at 48 to 96 hours, and making correlates between the progression of those biomarkers, and then relating that to then downstream assessments with where we're looking at whether it's Hammersmith or Bailey. We've spoken about the risk that this has to small to middle enterprise biotech companies as well, but there's also a risk to the families and to the patients to have these long protracted clinical trials as well. So it's really just about being able to have early stratification of these data sets for these clinical trials.
And the access to the natural history data, right? This is why Inc. and, NBIC, this new injury task force, I'm really hopeful that, we'll have even more. We've got access to a significant amount of historical data that is, helping to form a backdrop of our smallish clinical trial. And that is one way to, mitigate the risk in the long term, and the challenge of having larger trials. We're using some natural history, but the data, I was very struck by the work that's already been done with the software, and I hope we can push that along and continue to build out the data sets here in neonates. Be wonderful.
And maybe Adam, if you can combine, I've heard a couple of things that are interesting that you've said that triggered additional questions. Obviously, from obviously your experience and has been very valuable with planning around EEG and MRI. I mean, do you have any gut feeling of like where you feel gaps are? And Janet, feel free to chime in here too. And then I guess the other thing I heard that you say is you're hinting to some composite like biomarker that might be usable, and that might go with the real estate question. But do you have any thoughts about that?
Yeah, I guess in terms of the clinical gaps, we sort of feel that there are two, and primarily it's around the standardization of the assessments as well as the context of use. Is your drug looking at reducing inflammation in the brain? Is it looking at a neuroprotectant? That's going to drive your biomarker question. And that then drives what you're going to be assessing, whether it's MRI or blood-based biomarkers or, MRS, basal ganglia thalamus. And then if we're looking at the sort of the stratification and the sort of the harmonization across that data capture, which is where one of the big gaps is, especially if we're looking at MRI, is making sure that those scans are acquired using the same scan metrics across different platforms of scanners, whether you're looking at Siemens, Philips, or GE. None of them like to share anything with anybody. So it's about having the expertise to make sure there's no covariates being introduced with, your coil arrays or things like that. And then if you're looking at EEG and, building that out, it's being able to, take away, not completely remove the human aspect, but just to try and smooth over the assessment of that human aspect, and also build out reducing a lot of the noise around EEG. And, if we're looking at what's the signal of EEG doing during rewarming, we know that rewarming itself increases a lot of background variation within those signals. So it's really utilizing a number of different points of those data sets and combining them into a composite score, and looking at sort of exploratory biomarkers where we're taking a combination of these different biomarkers. Considering the presentation is so heterogeneous, we need to be able to map that to what we're seeing in the infants.
I mean, Janet, would you comment a little bit more? I mean, some of the things that I think that were said there that maybe from a clinician side really are additional thoughtfulness that could be added to the conversation. One of the things in operation trials like this, they're not easy to do. And so like, do you have any recommendations or thoughts about timing and essential points and how to, like, if you were talking to sponsors or other stakeholders interested in this, like what learnings could you share with respect to those things? Because the rewarming question for me is like, do you do it during deworming? Do you do it afterwards? Is it, how early is
You
enough?
mean like the MRI?
Yeah.
Yeah.
Either MRI or EEG.
It would depend. So the EEG, I think a lot of centers would continue EEG monitoring throughout hypothermia and rewarming because of the risk of recurrent seizures during rewarming. And so a lot of folks would do it during that period if it's at least moderate to severe HIE. Most people agree that if it looks mild or normal, that you can stop EEG monitoring earlier. And so that would be a challenge if you were going to include that. If you're talking about timing of MRI, I mean, I think the most important thing is to get everybody to do it at the same time, right? In the same way. And so, early on, we were getting MRIs a little bit, when we first started providing hypothermia clinically, we sometimes got MRIs a little bit earlier, trying to sort of hit that nadir of restricted diffusion, which is actually somewhere around 48 to 72 hours. But then feasibly, it just became so much easier to do it a day or two later, and nobody was like too excited about, well, it's slightly less optimal, but it's a million times easier. And so I think being able to harmonize what you do research-wise with what people are actually doing clinically and the way they're used to doing it is honestly the more practical way to go about it. And I don't think it's wrong,
Adam, do you have anything to add to that at all?
I am just sort of echoing the general sentiment as well. And, when we're wanting to develop or bring some of these novel biomarkers into the clinic, it's really understanding what the clinical landscape is. We don't want to reinvent the wheel or have to change clinical practice in the centers. And it goes back to that harmonization again. And if you're looking at multiple sites enrolling into a clinical trial, it's having that harmonization across the sites, which means having the sponsor across every single one of those sites and an investigative lead at each one of those sites so you can drive that harmonization. But it takes time, it's costly, but at the end of the day, it's generally just what has to be done in the current landscape.
Strength of having groups like that Betsy mentioned, like the Newborn Brain Society is, working on this common data elements. The strength of having people work on these sort of common data elements, common protocols, how do we do things? The more sites that do things in a similar way, the easier it's going to be to add those research studies onto clinical practice. So I think that that's something that people are trying to move towards in many places. And certainly for HIE, where there's been, honestly, so much variability. And then, nobody worried about HIE being a heterogeneous disorder before there was hypothermia. But then once hypothermia came along, it turned out we were just schooling all kinds of folks. So, it just becomes that much more important to have a more standardized approach and thought process to how do we approach these kids and sort out who's an orange and who's an apple.
Yeah, this takes an enormous amount of discipline, by the way, on the part of the sponsors, because we see them screening all different babies, but we have to stay strict on the NICHD criteria. We have to stay strict on, they have to get cooling starting at 6 hours. So there's, it's got strict criteria, and it's really just to try to keep as much of the variability out of the picture. And then also to your point about the MRIs, I mean, we actually do the MRIs at the times that the clinicians, 4 days, 12 days, and then we have one more that we do later on just for sort of additional information. But, we obviously engaged with lots of clinicians about how to create the protocol. But probably most importantly, I think, was that we engaged with Hope for HIE very, very early on in our protocol and in our development of the plan. And Betsy and her organization, played a really important role in developing how we go forward with our long-term part of the study. And we have an assessment on quality of life, which, as you may know, there was nothing really that well developed for newborns. It starts at about age 2, where people start, doing quality of life studies. So we actually created one on our own, and in conjunction with Betsy and her team, they did focus groups for us. And this has been, and we implemented the tool as a part of our clinical study, as part of our validation of it. And I hope one day it'll be fully validated for everyone's use. But it seems to be a really, really interesting tool. But it does take, sponsors, asking physicians, how do you normally operate, and how can we minimize the changes to your normal operation in the NICU to do the study, and then, of course, to get the feedback from the families who matter most.
Betsy, do you want to elaborate more on the collaboration and how you work with industry?
Absolutely. Yeah. So this has been, our first. We're collaborating with many of you now and others, both on the academic and industry side. But, Rialta has been a fun partner because we have kind of grown together as organizations. And we, one of their core values is, elevating the patient experience, and has been, very receptive as we come in and say things that they might not have wanted us to say, or that, that might not have been known, or things that we've been, chomping at the bit to elevate for many years that are articulated by our families, and things that our families have, talked about why they've opted out of a trial, or why they, at 6 months or 2 years, they don't want to go back, and what we can do to work together to make sure that, we keep all the babies as many as we can enrolled in the studies, because we know that these cohorts are small. We know that every bit of data is critically important. And so I think that's where, support really meets the rubber in the road of an integrated approach, holistic approach, looking at the science, looking at the family support, the feasibility. And again, these are all, I feel like there is nothing easy with HIE, right? It's so complex. We all know this. It's easy. We're in the weeds, but we can't let that, stop us from doing these hard things. We can do hard things. So. And we have a historical community and people that are around these tables that have been a part of this amazing legacy to get us to where we are right now.
And I guess the other question I would ask, I'm kind of trying to build on the theme of building clinical trials a bit, but from Anne's perspective, as you're thinking about biomarker development in both either the context of a program or outside of that in like a non-competitive space, do you have any advice as people are approaching this?
I mean, I think I've touched on some of these things already, but, again, it's context of use, context of use, context of use. So I think there's definitely a role, there's a need to expedite treatments. And I think there's always this tension between, you know, expediting drug development. Betsy and I've had a lot of conversations around this too. All of those things, when you start talking to families, it's, you know, the 2 years not enough, and we want the cognitive outcomes and seizures and epilepsy, and, you know, what happens at 7 years that we didn't see at 2 years, and all of those kinds of things. So there's this balance between, you know, getting drugs through the pipeline, but also keeping, you know, these other longer term or longitudinal endpoints in mind and their importance. So that's why when we talk about biomarkers, that's why I'm going to go back to, you know, there is a needed role and an absolute, you know, it's a imperative that we expedite things and incorporate these into the pipeline. And then what specific biomarker or where is the part where I think the work needs to be done, and where hopefully this group will focus. And as they take on a biomarker, I think those are the conversations, right? So, you know, is this in, you know, are the blood-based biomarkers, the PD endpoints, going to help us get from, you know, early phase to phase 2, and then these short-term endpoints, you know, getting us faster through the steps. And then when we get to the point where we're talking about registrational trials or reasonably likely surrogate endpoints, then again, that evidence base hopefully can be grown through some of these studies as we incorporate them, at least initially as exploratory endpoints, but eventually to the point where the evidence base is strong enough for us to say, you know, we can hang our hat here and say that this drug works from an approval standpoint.
Okay, I'm going to switch to questions from the audience. We've got a lot of experience of people. John, do you have a comment? John has a comment of everything. I was going to look at the audience and like, you do HIE, you do HIE.
Well, this has been a great discussion, and I can't tell you how delighted I am. Well, we have 3 companies up there that are interested in HIE. I mean, we really need the help. You know, we've just had therapeutic hypothermia, you know, that has a number needed to treat, sort of 1 in 11. And I recently took a lot of heat from a referring doctor who sent a baby in for cooling, and I didn't do it because the mother was preeclamptic and the baby's magnesium level was 6. And so once the baby's magnesium went away, the kid woke up and was totally fine. I mean, we're cooling everything now. And I don't know that, I call it mission creep. But I'm a consultant for the Cool Prime trial, which is cooling for mild HIE. That's something you folks hopefully will consider in all your drug development programs, because cooling affects the metabolism of so many drugs and makes it much more complicated. I think Janet was right with saying that some of these blood biomarkers, you can't really use CSF because we rarely tap the kids when they're that sick because it can be worse. But certainly some of the blood biomarkers, if you could use them and correlate it and say, well, if their myeloperoxidase level is above this, then that correlates with MRI findings or with later findings on Bailey's at 2 years of age. And for those of us that still work in the follow-up programs, I can't tell you how many kids I had that had what we would call a bad-looking MRI who come in with a very mild left-sided weakness, but are otherwise running down the halls and saying, hi, doctor, when I'm, you know, following them up. So that's what makes me nervous about using some of, and the MRIs we're not even getting until 3 or 4 days of age when the cooling's over. That's too late for you folks. You have to start your drug much earlier than that. So, you know, most of the time we'll get a head ultrasound when the kids are being cooled, but we won't do MRIs until they're rewarmed. So it makes it much more complicated. And some of these lesions, like strokes and other things, aren't showing up within the first few days. It's only later that you'll even see something even on MRI. So again, it makes it more complicated. And then that's why I think Anne and others in the regulatory agencies still sort of hold on to the Baileys, even though they're not perfect. You know, it's really only because we haven't given them anything better earlier. If we can, then I think there's a lot of opportunities.
Working on it for sure.
Yeah.
No, I think an important comment about that though is, we do the MRI at 4 days, but the drug infusion, we start infusing the drug at 10 hours of life. So they've already been on the drug. So we don't get a before and after MRI. We can't do it, right? I mean, it's impossible to get one, next to impossible. So we get the 4 day and the 12 day, and then out 42 days, actually, we get another MRI to look at maybe trajectory. And so those, that's the only way we can do it. But I think the other point Janet made was really interesting too about real estate. And this is why we use the wiki scores. There's been a lot of different scoring methods. They've evolved over time. And to the point where, I mean, we felt that the wiki scoring system seemed to be, per the literature, the most advanced and, picks out all different areas of the brain. And so that's the best we have. And that's what we're doing. So the hardest thing is, we have small numbers. These babies come in and few and far between, and we have strict criteria. We have terrific sites that are working diligently to, get babies consented. And, that's another course challenge that was spoken about here. But I know Betsy has actually spoken directly with some of our PIs to guide them a little bit on how, how to approach some of these families. It's invaluable
Jason, you
effort on her part.
Yeah, just so we're studying white matter injury in premature infants, so we're kind of outside of the cooling kind of system, and so but nonetheless, I think there's a lot of learnings we can get and kind of being involved and thinking about how to move this forward. But again, one of our blood-based markers, we haven't had much luck at all, but we are hoping that we have some luck with a head ultrasound to be able to detect the white matter injury and then potentially be able to detect, we'll treat the baby and then perhaps be able to detect some improvement then at an MRI at term, and then do some follow-up with the Hammersmith and a couple of other measures in the first couple of months. And we're we see it as kind of a stepwise. Again, ultimately the 2-year Bailey is kind of it is the gold standard as of yet. Pros and cons, but it is. But we're hoping that we can have some additional measures before then that will help us be able to predict either rates of cerebral palsy or rates of motor impairment and give us a signal. And just to echo, I think I think there's going to be stepwise progression. It's how do we move this forward, do some exploratory endpoints in our study, and see if we can build on the data that we're all generating. And I think that's one of the great benefits of working together with Inc. and the other groups, is to be able to test these things out and share the data and see if we can move it forward to. And just the last comment is we've been talking with our investors recently, and they, it was interesting talking, because we always thought, oh, we're saving babies. Why wouldn't anyone want to do that? Everybody's got to do that. But they came at it with, and it's kind of this financial mindset of like, well, there's lots of other opportunities. We can invest in oncology or autoimmune or other areas. And so they're thinking about the opportunity cost of those investment dollars. And so kind of the more we can do within kind of the neonatal space is to have these, diagnostic biomarkers or efficacy biomarkers. I think that'll help make it more attractive for investment and be able to continue to move us forward and get more products developed.
Yeah, but you're investing in a neonate who lived 70 years versus investing in an 85-year-old who maybe has 2 or 5 years. So
That's the investments.
You got to bring it along, John. We got to bring it to our next meeting.
John, spokesman, John, and little sound bites.
I just want to say a very big thank you to Betsy for being patient as Inc. pulled together the HIA side of things. I just want to thank you. You're a wonderful advocate. And I know it's been a bit of a road. Why are we talking about preemies only? I understand, be patient, and look at where we are. So I'm just super proud of you. You really are a force to be reckoned with. So yeah, thank you.
You got anything, Maren? Yeah, got anything? You're good. Okay, I thought you'd be on there. The only other question is, we have lots of jurisdictions represented here. I mean, obviously, we've been talking at the context of, FDA and obviously probably a little bit US-centric approach. Is there anyone in the audience wants to comment to their experience, like Carl or some of our Japanese representatives, about their experience and how this might fit into care?
I'm not sure if I represent Europe, anyhow. But
please Belgium.
But in the absence of others, yeah, sometimes things happen, right? I only would like to share that we recently finalized the albino study, or we stopped in intermediate, and we learned also a lot over there. The complexity, I'm not sure if this over here in the US, but Europe have a lot of different countries, different cultures also to look on consent procedures, deferred consent, short verbal consent, and it has been alluded, I think, in the previous talk, these mothers are not there in generally. They, even if they have consented, they do not know it the day afterwards. Genuine. So it's a construct that we want to work with, but it has shortages anyhow. And what we have seen in different countries, like for instance, Poland never accepted the study. It was unethical. While other countries said you need shared, written, informed consent from both parents. So de facto, they killed the study. Others accepted deferred consent. So you have a lot of different aspects, and the only way that I would like to look at it is that somehow you still will have to respect the cultural, the social background or whatever of a given community. And some are winners in the system and others are not winners in the system. And indeed, a lot of babies have been included in the Cooled studies, and it was because parents believed that it was useful to contribute. And yeah, you have to accept that not everybody will be ready, willing, or able to have their baby included in the trial. So that's the first point. The second point is MRI. It's a mess because everybody has a different equipment and different settings, and so that's difficult. EEG, indeed, you can have central readings, you can have whatever, but it has a lot of issues in the European context thanks to GDPR. That was ironic. So yeah, I recognize a lot of things that have been said, perhaps with some European flavor. And the only thing that I would like to add to your comment is we are working, as you know, on a PBK model for asphyxiated babies so that we can predict drug exposure. And the interesting thing is not everything goes down. Some isoenzymes are upregulated, and so potential toxicity or clearance could be higher in newborns without following asphyxia. So it's, again, not easy, but it's fun, and we are making progress, and we have to continue to try to do so. Japan?
We're going to go to the UN here.
Well, definitely I'm not representing Japan because I'm not even a neonatologist, although I have some experience in neonatology. I'm a little hesitant to talk about this, but when there was a discussion about master protocol on neonatal seizures, Japanese neonatologists said, well, we don't usually take 24-hour EEGs. And I thought there are two sides. And sometimes for other clinical trials, not necessarily in neonatology, there's a new scores or whatever the evaluation method which is not commonly used in Japan, and that could be the hurdle to conduct global studies. Well, Japanese neonatologists and physicians should try to introduce whatever the new methodology as quickly as possible. That's one side. But there's a discussion as well, if it cannot be done in Japan, it probably cannot be done in many developing countries. And I'm hoping that we can find out something feasible in many different countries so that we can do global studies more easily.
Okay. Well, if we don't have additional questions, we'll close the panel because they're flashing signs at me, which I think fine. Okay. Okay. Thank you for the panel.
It's everyone.